Hello, my name is Lali Gomez, and my student presentation seminar is on CRISPR-Cas9 DNA editing efficiency increase due to DNA ligase 4 inhibitor SCR7 favoring homology directed repair over non-homology and joining. So CRISPR-Cas9 is one of the most powerful genetic tools as of today, and this is because of its ability to change the DNA sequence of an organism. So this involves the deletion, the insertion, the modification of the genome at a specific site in a DNA sequence. So CRISPR is a clustered regularly interspaced short palindromic repeats. And this is repeating DNA sequences derived from bacteriophages. And these sequences can be used as a bacterial defense mechanism, like an immunity system. And Cas9 is an associate protein 9. It's an endonuclease which enables double-stranded breaks. So overall, a short sentence, CRISPR-Cas9 it has the guide RNA to help target a specific DNA sequence, and then which is then cleaved by Cas9, inducing a double-stranded break, and then it is repaired. But the repair part is where the genome editing comes in, where the modification of insertion, deletion, modification comes in. The discovery and the history of CRISPR. So Asuro Nakata and his group of researchers in Japan discovered these short repeated sequences of the DNA and the palindromic repeats in E. coli. They found it in E. coli. They didn't know what it was, they just know they found this. But it was until 1990 where Francisco discovered the function of it and named it CRISPR. However, the mechanism of it was still a mystery. They didn't know. It was until 2007, using the bacterium Streptococcus pyogenesis, it was discovered that CRISPR has a bacterial defense mechanism. And this mechanism is when the bacteria is infected by a virus, Cas9, and Cas9 snips off a piece of the viral DNA known as a protospacer adjacent motif, and this fragment is stored in the bacterial genome, and this viral fragment are placed between palindromic repeats. Thus, when the bacteria can then recognize and kill it with the Cas9. In 2012, Donna and Carpentier discovered that CRISPR-Cas9 could potentially edit has the ability to edit the DNA sequence. So, CRISPR-Cas9 mechanism. There's 3 steps involved: recognition, cleavage, and repair. So, recognition. CRISPR-Cas9 has to bind to a single guide RNA, and this single guide RNA consists of the CRRNA and the track with the cRNA, which helps Cas9 recognize the sequence, and then track, which is over here, track RNA, which helps bind the sequence to Cas9. So this recognizes the sequence through the 5' cRNA complementary base pair. So, and then cleavage, it cuts a particular genome at upstream of PAM. Up here, you see where PAM is, and remember PAM is the viral fragments in the genome. And then cleavage, it cuts it causes a double-stranded break, and this is the repair part, and there's 2 repair pathways. So CRISPR-Cas9 double-stranded break repair, the two pathways involves homology direct recombination and non-homologous end joining. The two, NHEJ is faster, it's more common, it's the first pathway for after the double-stranded break. However, it is error prone, it's imprecise, it causes indels, mutations, it causes frame-shift mutations, gene knockout, as you can see here. While homology direct recombination requires a DNA donor template for its repair. And it does insert new genes, fragments of genetic material, gene knock-in, and it is more efficient. But the problem is that it is not what comes first. Comes first is the NHGA repair, causing the mutations, the indels. Okay, out of this is the two pathways, the homology direct repair and the NHEJ repair. But the important thing to get from this slide is that NHGA requires a DNA ligase 4 for it to work, while homology direct repair does not. Because as you see, these are all the proteins involved, but ligase 4 is what connects the two ligases, the two double-stranded ends to ligate and to go back together. It helps it. While double-stranded break needs a template for its repair to happen. Okay. SCR7 is an inhibitor of the repair pathway of NHEJ. It's a heteroaromatic compound, and it prevents DNA ligase 4 from binding to DNA and by acting as a competitive inhibitor at the ligase DNA binding domain. So here is an image of representing that of the process. So CRISPR-Cas9 happens, the double-stranded break happens, and then as you can see, in a normal situation, NHEJ is favored. It weighs heavier, it's the first thing to go, it's faster. So double-stranded repair goes in process. However, with SCR7, it binds the proteins, it binds the NHEJ proteins to bind. It's blocked, so it shifts the balance to homology repair, which is what we want, as it's more precise, it's more efficient. So here is just the different forms of SCR7, where parental SCR7 can be converted to SCR7 cyclase, which is by autocyclation, which is the formation of rings, and that can be then turned into SCR7 pyrazine by dehydrogenation, which is the removal of a hydrogen. Okay, how was it discovered that that inhibiting ligase 4 improves homology repair over NHEJ? So there's a study done in Drosophila, which is fruit flies. They were co-injected with zinc finger nuclease and a circular donor DNA in ligase 4 deficient flies. So these flies didn't have ligase 4. and they noticed that there was an increase in homology repair. As you can see in this table, the ones with ligase 4 didn't really take the process of homology repair. However, the ones that didn't have ligase 4 showed a major increase in homology repair over NHEJ. They didn't take that repair pathway. So this is why it was then led to believe that ligase 4 was the key to increasing CRISPR-Cas9 DNA editing. So this is why SCR7 is being used in this experiment. Okay, so why is CRISPR-Cas9 important? Why do we want to improve it? Well, we want to improve it because it rewrites genetic code in the DNA. We're able to use it for cancer treatment, such as the breast, the colon, the liver, and these are the processes of which it can help in. So why wouldn't we want it to improve it? And there has been successful treatment in using CRISPR-Cas9 in genetic diseases such as the sickle cell disease, diabetes type 1. Furthermore, the applications of CRISPR-Cas9, this is why it's further important, and why scientists and why research is being done to improve CRISPR-Cas9, because what it has on cell and gene therapies, it has the potential to cure diseases like HIV. Who doesn't want a cure for a disease? And diagnostics, it can be a potential diagnostics, as it was used. CRISPR-Cas9 was used for the coronavirus. Agriculture, for the crops, for the food, disease and drought resistant, and it is a great disease. It's greatly used in disease models and for disease models in animal models. It's faster, cheaper method in the lab. So my hypothesis is SCR7 suppresses NHEJ by inhibiting DNA ligase 4, not required by non-homology repair, increasing CRISPR-Cas9 DNA editing efficiency. Homology repair percentages after treated with SCR7 in zebrafish. So zebrafish embryos were injected with Cas9 protein or Cas9 mRNA, and then they were treated with SCR7, and then DMSO is just the control. And as you know, homology repair requires a donor template. So single-stranded DNA, double single DNA, and plasmid were used as the donor templates, and they were used to induce the substitution of phosphorylated serine residue with an adenine at position 82 of Y box binding protein 1. After they were treated with SCR7, you can see a significant increase. You can see a significant improvement of the homology repair efficiency in the zebrafish embryos for all three of the donors, because as you can see, these are the ones with the control. So you're comparing them, right? It increased. You see an increase. You see an increase. You see an increase. And I know I'm covering it, but you see an increase. So it improved. The CR7 improved homology. The homology repair 55%, 25%, and 13%. Here is 55, 25, and 13. While the controls were 15, 10, and 5. So the plasmid showed the highest efficiency of point mutation and the presence of SCR7. Okay, the improvement of homology repair efficiency in xenopus. Okay, so xenopus oocytes were treated with CR7 after they were injected with CRISPR-Cas9 for knock-in for beta-catenin, which is encoded by the gene CTNNB1, which is involved, which encodes beta-catenin, which is involved in like the WNT pathway, which regulates cellular functions like proliferation, which is not important, but just what the gene is. And as you can see in figure G, there's without SCR7 and with CR7, and as you can see, there's a major improvement in the knock-in for the gene beta-catenin. As you can actually see in SCR7, the fragments on the Western plot. And then on figure H, xenopus tadpoles had treated with SCR7 shows the percentage of homology repair, and both of these in the tadpoles, both genes were independent. These independent genes were used in seeing how the homology repair, and it shows that there's an average increase of 7.4, which with no homology repair, to a 22.1% increase of homology repair. However, SCR7 showed was toxic and lethal in this case with the embryo injection. As you can see, they're just 0. Effects of CR7 in rabbit embryos. So rabbit embryos were injected with Cas9 for RLL-EGFP knock-in. So RLL is a useful location for inserting a gene without affecting other genes around. And EGFP is like an enhanced green fluorescent protein. It's like a reporter protein that exhibits green when it's exposed to light. It was shown, and they were treated with SCR7. However, there was no significant effects of the overall knock-in efficiency, because as you can see, as the concentration of CR7 increased, there was minimum effects. So there was 7.7 in the control at 20, and it went up to 9.4, but there was no dramatic increase in the efficiency. Oh, and PR7 pyrazine was used in this experiment, in this independent study. SCR7 in porcelain fetal fibroblasts. So in figure A, in this one figure, SCR7 was used to determine to see the cell viability using MTT assays. MTT assays is a colorimetric assess for assessing cell metabolic. So these porcelain fetal fibroblasts were treated with SCR7 for 48 hours, and there was minimum effects. SCR7 is one in blue, and covering it with the imagery, and this is the control, and as you can see, there was minimum decrease. There's minimum effects. On figure B, on this side, GFP reported plasmid was digested with Hindle-11 to evaluate the homology repair efficiency in porcelain fetal fibroblasts by flow cytometry, and which analyzes single cells as they flow past or multiple lasers. And they did improve by up twofold compared with untreated. As you can see, the control is at 5.6, and then the CR7 increased to 11.2. The enhancement of insertion repair at AAVS1 locus by SCR treatment in MCF7, which is breast cell lines, and HCT116, which is colon cell lines. Both cell lines were treated with SCR7 treatment, and as you can see, these arrows are the original uncleaved DNA fragments, and these plus signs are SCR7. So SCR7 didn't have it, had one CR7 and then didn't have it, right? And as you can see, with the one with all CR7, you can barely see it, but it does recognize and cleave, and did homology repair did happen, as you can see. And as you can see, it went from 1.1, as you can see, really light, to 4.3% increase. And this is in colon cell lines. And in breast cell lines, as you can see, increased from 2.7 to 6.6%. And this was done by restricted fragment length polymorphism assay, which is helps differentiate DNA sequences at specific sites by the different sizes in the DNA fragments, as you can see. SCR7 enhanced mutation correction efficiency in the breast cell lines. So they had a silent mutation that resulted in low green fluorescent protein, right? Right, this is the mutation. This is the wild type, a lot of it, and this is the mutation. And this figure was done by flow cytometric analysis, and this is analysis of single cells that pass through multiple layers, as mentioned before. And as you can see, once they were added with CR7, it enhanced the mutation correction. It started increasing the levels of green fluorescent protein, and it increased to 6.6%. Conclusions and discussions. So while the efficiency of CRISPR-Cas9 increased, genome editing increased, the genome editing, the DNA editing increased due to CR7 inhibition of DNA ligase 4 as it favors HR over NHEJ. There is additional work necessary to confirm the effects of CR7 on increasing homology repair efficiency, as there's evidence from the experiments, from the results, that there was minimal effects on some, while in some experiments there was huge improvements. And so there still needs to be further researched and research to be done. These are my references. Thank you.